Today we have a very special today we have a very special guest with us, Dr. Mrinalini Damle from one of the prestigious institutes of India situated at Pune. She's a professor working over there and has an academic research experience of over 24 years. My gosh, this is a wonderful time, and we are lucky enough to have her on this platform to, interact with the people who are working on this particular aspect of using HPTLC for academic research. And incidentally, when we approached her for this event, she has been so helpful to students and everybody that immediately she took up and she's very senior and a lot of experience. She has areas of interest in analytical bioanalytical method development, validation, stability testing of drugs and pharmaceuticals, herbal drugs standardization, publications. She has a lot of publications in which she has been reviewer type 11 publications, national research publications 23, international research publications 135, H index publications 16, I-10 index of 26, citations 830. So, it's a huge experience of years of work today. And she has many presentations done as an author, as a co-author, and we are really fortunate enough to have her on this platform interacting with all of young scientists who are working on HPTLC on the aspect of standardization and the others which I stated. So, without further extending my speech, I will now invite ma'am for this wonderful session. This will definitely take us to the next level, and we are so again once again thankful to all of you. Please, if you have questions and answers, we can take it at the end of the session, or you can type it in the Q&A box from our team, and ma'am, we will get those answered and revert back to you. Those questions which are not taken up in the session, we will be reverting back to you on email. So don't worry about that. With this note, I will now invite ma'am for her wonderful lecture, and we look forward for a wonderful experience. Thank you, ma'am. Over to you. All the best. Thank you, ma'am.
Thank you so much, Sekar sir. Am I audible? Can you please confirm?
Yes, ma'am.
Yes, ma'am.
Thank
Absolutely.
you so much. At the outset, I would definitely like to thank Anchrom for inviting me. It's always a pleasure interacting with the researchers who are budding researchers and maybe my colleagues all over India who are in academics doing a similar kind of research. And it's always good to learn from each other. So when we say we publish our work in a peer-reviewed journal, I definitely welcome the suggestions by my peers, seniors, juniors equally. So I'm always open to suggestions for improvement. So today it's, as Saikat sir put it, my experience of 22 years in academics and 8 more years before that was in pharmaceutical industry. I suppose that my industrial experience has helped me a lot during my teaching career. And as our college started with PG course, that is MPharm, I was like really happy that whatever I had learned during my MPharm days, during my PhD work, has helped me simply because it was hands-on. So what we learn in class and what we really practice in lab, I think both put together help us in developing our career further. And I hope that whatever I discuss with you today, I will still put it as discuss, because whatever work was done by my PG students, MPharm students has been published in good peer-reviewed journals. I'll be sharing some of those because all of those papers are open access. So without a copyright issue, I think I'll be able to share the contents with you. And since it is open access, all of you can refer it anytime later also. Your queries, your suggestions are most welcome. So let me begin. Let me try presenting. I hope you are able to see the first slide, which is HPTLC in academic research.
Ma'am, we are able to see the paper which you have opened
Paper
on
and
PDF.
not the PPT.
Yep, and not PPT. Maybe if you can just put in the presentation mode.
Can you or not still?
Yeah, we
can
can
see it,
see.
ma'am.
Yeah,
You
thank you.
can see the
Yeah.
PPT now?
Yes,
Okay.
ma'am.
So good morning all. Uh, I would start with my topic, which is HPTLC in academic research. I know that academic research, when we say as a teacher in pharmaceutical or pharmacy college, my objectives have always been that I should be able to impart knowledge as well as practical skill to all of my students. So then my students are industry ready. This is a term that I have been hearing recently as we go for accreditations. It could be NAC or NBA. The objective, as they call it, of our teaching is making our students industry ready. And that I think cannot be overlooked, because when we teach you in theory class, we expect you to apply those in your practical lab, acquire the practical skills, which we call it as hands-on experience, and that makes you ready for industry. Since I have been guiding students from quality assurance branch, I definitely make it a point to see to it that my students are aware of various regulatory requirements. It could start with something as simple as a pharmacopoeia to knowing US FDA 21 CFR different parts, knowing the concepts of GMP, CGMP, GLP, various guidelines like ICH and PICS, and this there is no full stop. These guidelines are essential if to be known and understood, to be ready to apply, because when you join industry, maybe immediately after your PG, you should feel confident enough that you have applied. When you develop a method, you have validated it, you know the ICH guidelines ins and out. There is a concept of data integrity, and there is a lot of talk and discussion, I'm sure, during pandemic period when all of us were uh, I mean, I must say fortunate to listen to so many webinars. Data integrity came to forth, and this is a concept that every budding student who is ready to make a career in industry should know about it. So with these objectives in mind, we go ahead and ask our students to take up their research. And if I may divide the research into two basic types, it could be like basic and applied research. At least my experience so far has been in the second aspect, which is applied research, because it's the pharmacy students that I be interacting with. So in applied research for pharmacy, it is in our college, we do have a system wherein even UG, B.Pharm students are encouraged to take up short-term research projects. So my research in my college starts with even UG. So my UG students, those interested, of course, not it's not compulsory in any way. So interested students do take up research projects. They're short term, something they do without compromising their. I think we are talking from home, so we'll have to bear for some time if there are hawkers outside. I hope you can still hear me. Okay. So going from UG, some students do take up research at UG level. At PG, definitely in farm, they have 2 semesters to take up research work, and PhD, I think all of us know that it's quite extended kind of a research. Now, coming to today's topic, HPTLC, a type of planar chromatography. We know definitely that chromatography in pharmaceuticals is highly important It has variety of applications, something that you could discuss on and on and on. But today our topic being limited to planar chromatography, especially HPTLC, let us see that even planar chromatography can be used for both the purposes, preparative as well as analytical. But you might agree that um, the when it comes to preparative chromatography, generally it is column that will be given, column chromatography will be given a preference. But I would say that when it comes to analytical kind of a work, equally column and planar chromatography go hand in hand. Many of my students have developed a method using both the techniques, even compared it using statistical methods, and they have got, acquired good enough confidence before entering R&D or QC department in industry. So, planar chromatography with respect to analytical aspects is something I wish to discuss in detail today. When it comes to analytical method development, um, the process should start with knowing the physicochemical properties of the API that you have at hand. Unless you know unless you have searched the literature for such physicochemical properties, I don't think my next step of even development could start on a proper basis. So thorough literature survey, knowing the physicochemical properties, should help you to start your work on a strong background, and then you start with the method development. In planar chromatography, yes, we know that in TLC, we are interested in getting a peak with a good RF value. You could say good would mean probably something between zero point 0.2 to zero point 0.8. Now, this is traditionally speaking, zero point 0.2 to zero point 0.8. You're going to see ahead in my presentation that yes, there are some situations wherein you could have a RF value which is extending beyond these ideal limits, but then under certain situations you do accept it. So uh, having an RF value which is acceptable, having a peak shape which is prim and proper, symmetrical is the word I would prefer. So having a symmetrical peak, having RF value within the acceptable range, and moreover, if you have many components to resolve, having a good resolution, as we call it, baseline resolution would be your objective. Then what is optimize? Optimizing could be not settling for the first okay kind of resolution, trying to go for best, which is optimization. And then we know that there are many softwares available. Many of you must have heard of or even practiced quality by design. Design of experiment is the software the students at our college have been using, so as to have dependent independent variables, and then having a practice of optimizing the method. Today may not be the right time to talk about what is QBD, but uh, I must at least mention it at optimization stage. Once you have your method optimized, you are ready to validate it. And something that students definitely learn during their theory is ICH guidelines, ICH Q2, AR1, and then they must know all the parameters. They must know what each parameter means and how it can be really practiced. So when it is linearity and range, precision, accuracy, robustness, so on and so forth. For a regular analytical method, having discussed this in theory class, now is the right time to implement it. So method validation. So this is something that's something my students do get hands-on experience when it comes to academic research. Here I may mention that when it comes to bioanalytical method, the validation parameters are quite different than regular analytical method validation, and it is a bit tedious. There are too many samples that you need to handle, and it tests your patience many times. So method validation is nice to learn in theory class, but when it comes to implementation, yes, I suppose that's the right time to look for an opportunity to thoroughly understand the meaning of each and every validation parameter. Now let's come to what all are the areas under pharmaceutical research that HPTLC has been applied. Now, again, let me put it. Whatever your six headings that you can see listed below, I have restricted myself to only these six because this is what have been implemented in my lab at AISSMS College of Pharmacy. But this is not a limiting list. This is definitely not all that HPTLC can help you with. There are too many other applications, but since my talk will be restricted only to whatever has been done in the QA lab or central instrumentation lab at our college. So we have applied HPTLC for pharmaceutical method development for the following purposes. It could be identification, assay, standardization of various herbal drugs, development and validation of bioanalytical method, that is determination, estimation from plasma, urine, saliva, development and validation of stability indicating methods for bulk drugs and formulations, and definitely applying this analytical technique to study of dissolution samples. So these are various, areas which we have tapped so far. Uh, there are many more that we will definitely be taking up in near future. But I think let's start with the first one. We have used HPTLC many years ago for identifying a marker in the plant Anantamul. So Anantamul, the pharmacognosy name would be Hemidesmus indicus. And one of my students, Rahul, had used it for extraction and isolation. Can you please confirm if you can see the paper share?
Yes, ma'am, we can.
Thank you. So here you could see the title is HPTLC quantitation of 2-hydroxy-4-methoxybenzaldehyde in Hemidesmus indicus root powder and its extract. Now this paper was published way back in 2009 in Journal of Planar Chromatography. This work talks about isolation of a marker. This is the TLC densitogram, wherein you have the peak eluting at around 0.7. You can see the peak shape is symmetrical and the RF is within 0.2 to 0.8. Now this is acceptable limit. Once you have your active marker isolated, its TLC method developed, you would like to quantify this marker from the root powder that you could get commercially. You could purchase it, you could extract it, and we were to find out how much is the percentage of this marker in the root powder. And once you get another peak at similar RF, we could easily confirm that this is the same marker by going for something called as spectral identification. So you go for UV spectral determination. If you have two peaks at the same RF value from two different racks, you go for spectral comparison of the UV spectra for these two peaks. And this is how we could confirm that or identify, or I could put it, the correct technical word could be confirming the identification of the marker, which is you could see the overlapping UV spectra, almost perfect match here on the right side and slightly differing on the left side. So here it says the UV spectrum of isolated marker compound, and the other one is UV spectrum of a compound from the root powder. So not only will the RF value tell me that this could be the same compound, I have to go and confirm its UV spectrum to say that yes, the isolated marker and the one found in the root powder, they are probably the same. So this is regarding identification of a marker, the first application. Next, the very common application is assay. Assay using chromatography, that is TLC, HPTLC. And we know that when I'm using the term just TLC, it is maybe for identification, qualitative applications, And if I have to use the same TLC for quantitative applications, yes, I have to depend only on the automated system, which is HPTLC. We have been using the common TLC plates for this, but the quantification is possible simply because it is the application is automatic, the scanning is by a scanner, and it's not simply visualization under UV light. So for assay using TLC, what could be the challenging factors is when you have more than one sample, that is multiple actives in your sample, that's something that could be challenging for any analyst. If you have a skewed proportion of two actives in your sample, uh, let me explain what I mean by a skewed proportion. Suppose out of the two actives, say for example, one of my students had worked on a combination of nebivolol, hydrochloride, and valsartan. The nebivolol in this combination is just 5 mg, and you have valsartan, which is 16 mg. So, sorry, it is 80 mg. So it is 16 times. So this is what I am referring to as a skewed proportion of the two actives. And you might wonder as to what does the dose have to do with TLC pattern is when you have two actives which are eluting close to one another in your optimized TLC mobile phase, if the proportion of one of the ingredients is very high as compared to the other, we can even think of one more, a very common combination of paracetamol 500 and some other drug, which is 2 mg, 5 mg. Now just see the ratio is not even 1:16. It is something like 1:100. And there comes a common problem that you have one peak as you take a sample, representative sample from this marketed formulation, you will find that these two come out in different ratios, and then having that peak resolution still maintained could be real challenge. Third could be the two drugs in combination having different lambda max values. Now in HPTLC, it's great help that the same plate you could scan at two different wavelengths without you having to do any other different sample preparation. Now that comes as a great help. So even if your drugs have different lambda max, yes, you can still go ahead and work on a HPTLC method. The next application, which is very, very popular, is when it comes to herbals. Herbal drugs, as we know, are always, you think of any plant, it always will contain number of constituents, number of components are present. And then as I was talking about, say, multiple actives, the resolution is a challenge. And when it comes to herbals with multiple constituents, it is even more challenging. So how do we go ahead with herbal standardization? It generally, we go for something like selecting one or two or at the most three markers, develop a method for those markers, and then try to quantify those from the herbal extract, the raw material that you have, and maybe extracted that, and then you could go ahead and quantify it. So let's see if we can have a look at one such example of marker quantitation. Here, my student Swapnil had worked on extract of onion peels, which contains an active ingredient, protocatechuic acid. So here he had extracted it, quantified it, and even evaluated hepatoprotective activity for the extract. Now let's look at the TLC pattern. Now this is a UV spectrum for the marker selected, which is protocatechuic acid. Now this is a prominent marker known to be present, known to be active, having anti-inflammatory and anti-inflammatory, that is activity. So we thought of checking the hepatoprotective action. So this is the UV spectrum of protocatechuic acid. He could develop the method for TLC, the ideal peak shape and the RF value. And then he could go ahead and extract it from the onion peels. As we know that it is a good idea to compare the UV spectrum from both the extract as well as the typical marker. So the similar shape might help us to get good enough confidence that the peak that we are tracking is the same as the marker. Now here in the 3D densitogram, you can definitely see, let me show the title. It says that it's a densitogram of protocatechoic acid. Track 2 to 6, the first few tracks, as you can see, are depicting the linearity of protocatechoic acid, and the tracks behind at the towards the other half are from onion peel extract. You can see that in the sample densitograms, there are a number of other peaks also present. And then there is a marker peak, which is quite in a good enough quantum. So then Swapnil could determine the content, he could quantify it, he could go for correlating the hepatoprotective activity and the content of protocatechoic acid. So luckily, now I may call it luckily, because in this case, the content of protocatechoic acid is quite large. As you can see, the other components are there, but then this peak is quite prominent. Now, it may not be so in all the cases, because depending on what solvent you choose to extract your actives, the content of active could be quite less in the extract that you have chosen, and then trying to resolve the peak of your marker from other too many constituents present could be taking up a lot of your time during optimization. During herbal standardization work, the work need not really stop at merely quantification and identification of active markers. You could go ahead and even study the stability pattern. So when we say that stability indicating method development is a common kind of a work taken up by many QA students, you could use it, apply it to even herbals, and say, let's see one example wherein stability studies have been done. See, this is some work by my student Pallavi. She had worked on HPTLC estimation and antibacterial effect of methanolic extract of Naxomica seeds. She had extracted it, the Naxomica seeds. We know that strychnine and brucine are two active principles here. We had studied the antibacterial activity of the extract to start with in the beginning. Once we could confirm that the extract does have a good activity, we thought of going for exposing this extract to accelerated storage conditions, and then let us check what happens to the content of these markers, whether they reduce with time with stress conditions like this, and whether even that antimicrobial activity, will that be also reducing with the storage conditions at accelerated storage. So in this paper, yes, here we have the structures of strychnine and brucine, the two markers which were identified, quantified, and then we will have a look at antimicrobial activity as well. So these were the two markers. Now, as you can see, the RF values are much lower than expected. But then we could settle for this because the work involved even having comparison with antimicrobial activity. So since it was a two-way track to compare the activity, in spite of too many trials, we did settle for this because the resolution was good enough. So this was the starting point, and then antimicrobial activity was tested. After initial first month, second month, third, and sixth month, as we call it, the activity tested was against E. coli and S. aureus. The methanolic extract was tested, and the conclusion of this paper was that though you have slight decline in the content of both the markers, there was not too much impact on the antimicrobial activity that was seen. So this shows that we could still go ahead and use this methanolic extract because the activity for which it is being used does not degrade with accelerated storage conditions. So this was a good work by Pallavi, wherein not only were the markers quantified, the extract was even exposed to accelerated storage conditions. And then we could compare the content of markers at the end of 6 months, compare it to the antimicrobial activity. So under herbals, we would say that HPTLC comes in as a great device to our help. Let me put in two more points here which go in favor of HPTLC, because in my lab there are a number of students who have worked on standardization of herbals using even column chromatography, that is HPLC. If I were to compare the two techniques, HPTLC versus HPLC, all of us know that both the techniques will have their own plus and minus points. Yes, that's true. But especially for herbals, wherein the constituents present are a variety of them, they come from different class of compounds, different polarities, different solubilities, and we have actually no control over the content of each, because the content from the raw material could vary with so many parameters. As we learn in pharmacognosy that the solvent used for extraction would matter. Different seasons also lead to different content of actives within herbals, that is raw materials that we procure. The location from where you procure your raw material also has an impact on content. Now, having said this, if there is a lot of uncertainty in the actives or different constituents present, different class of compounds present in the herbals, when you want to inject it onto HPLC column, we are not really sure whether all of those components would elute after some certain time from the column, and nothing would remain lodged onto column permanently. Now, you can see that there is a danger. If there are certain components which are having a great affinity for the stationary phase in your column, the type of column that you're using, cyno, phenyl, C8, C18. If there are some components which have a high affinity for the stationary phase, they tend to just cling to that column stationary phase and they may not elute out. Now, that's a serious problem in column chromatography. Something that you may not even realize that out of 100 components, maybe 97 have eluted. And when it comes to academic research, let me put it that there are practical constraints that you have, say a fixed number of students who are eager to take up research on certain chromatography technique. And I don't think any college will have more than 3 or 4 HPLC units with them. Again, I'm restricting myself to private pharmacy institutes in India. So I'm not talking about government funded institutes. So there is a big gap between the two. So in private institutes, my college has 3 HPLC units. I think there may not be many private pharmacy colleges that have even this number, all of them in working condition. So I'll come back to my point. So if you have a herbal constituent out of so many components present in it, if some of them get lodged onto column, it's a column life that is getting affected over the period of time, because accumulation of certain agents on the column permanently until you change the mobile phase, making it essential for that drug component to elute out, and for you to wait for long enough for this elution to occur. This may go on happening. And what's the advantage in TLC? The TLC, every plate is like a renewed stationary phase for us. The plates are not that costly. Compare the cost of HPLC column, which goes something like in 35,000 per column, to a plate which you could 20 by 20 plate you could get for something like 200. I mean, when you buy a box of 25 plates. So renewable stationary phase is a great advantage. So even if you have some components which did not elute with your mobile phase, you could think of washing those off with other mobile phases. You could go and confirm, have a look at your cleaned stationary phase once again. And in spite of using different mobile phases, washing solvents, if your plate does still have some components lodged onto it, I mean, you could easily afford to go for a new plate altogether. So herbal standardization, we would definitely prefer HPTLC anytime over HPLC, simply for this one factor that it will have, yes, the advantage remains that you have high throughput as the biggest advantage of HPTLC, that you can have number of samples tested in one plate. But at the same time, the biggest advantage under herbal standardization heading would still remain that every time a renewed surface of stationary phase is what we are looking for, and that's a possibility. So going ahead from herbal standardization to bioanalytical methods. Now, this is a common heading given to estimation of drugs from the different matrices of, say, human origin or animal origin. So when it is bioanalytical, the biological matrix is expected. The matrix most commonly tested is human plasma. It could be saliva and urine and CSF and many other fluids, but the very, very common one being human plasma. Now, when it comes to estimation from human plasma, the hurdles that come to forth is, number one is, what is the Cmax of the drug expected in plasma? All of us know that Cmax is the maximum concentration upon giving a therapeutic dose to humans, and this is available in literature. So if you know that Cmax of the drug that I'm planning to determine is something like 4 microgram per ml, that's quite handy to know this. I mean, knowing this very well, you can plan your work. This is a basic requirement to know your Cmax before you start any bioanalytical method development. In my lab, there have been number of methods developed on HPLC as well as on HPTLC. On HPLC, you will have an advantage that you can have a very, very longer runtime. It will help you to elute the samples which are not so easy to elute out. But again, similar to herbals, a common problem here is the plasma proteins. Though you will have number of sample pre-treatment methods, it could be simply protein precipitation or liquid-liquid extraction, or it could be solid phase extraction techniques. You can still expect some interferences from the matrix, and you will have the, yes, an opportunity to use a guard column in HPLC and try to avoid damage to your column. And on the same lines, as we talked about herbals, if you want to really protect your column and not have the guard columns wasted over the period of time, you could always go ahead and use HPTLC technique for this. So I would like to show you, say, one of the examples of determination of, say, ofloxacin. Now, Pranjali Ranavre, she had worked on estimation of ofloxacin from human plasma. The method developed was using some internal standard as is recommended for any bioanalytical technique, that you have internal standard which has similar extraction efficiency. They are generally required to ensure or give us confidence that our extraction plasma pre-treatment technique that we are using is working good enough. So the TLC, typical densitogram, as you can see here, the first one, as is labeled in figure number 3, is a densitogram of human plasma spiked with ofloxacin, and the quantum, that is amount that is spotted here, is 200 nanogram per band. As we know that some internal standard would be better, may not be mandatory, but better is having some internal standard. Now, when I say some internal standard, definitely a requirement is that it should get well resolved from the drug of your interest. It should not merge with any other peak. Like say some peaks you will see at the beginning of a densitogram, which are generally some components from plasma. So here we can see the internal standard which was chosen here has been well resolved, and this is what is expected in bioanalytical techniques. The method of extraction will help you to say confirm this or achieve this, that and here as you see here that in bioanalytical method validation, apart from routine validation parameters like accuracy and precision, you have the concept of stability to be tested for the sample, and then there are 5 types of stabilities that you need to test. That makes the number of samples many of them. So as compared to simple analytical method validation, the number of samples that you need to handle for bioanalytical method validation aspects become too many. And I'll come back to the advantage of high throughput for HPTLC, that you have too many samples to handle. There is no better way than having a plate, 20 by 20 plate, wherein you can have, say, good number of samples applied in one go. Your plate will develop in, say, at the most half an hour, and you are done with the content analysis of, let's say, 20 samples in 1 hour. Now contrast this with HPLC, typical runtime of say 10 minutes. You can imagine for 20 samples into 10 minutes, you can imagine its number of hours that you have to invest. And here high throughput advantage of HPTLC will anytime outweigh the HPLC considerations. So I may also go back to one more consideration here, that when it comes to sample handling, your plasma treatment, sample preparation will matter. How you are, extracting the sample will matter. So here we have a spike plasma, and it is being treated by a technique called as liquid extraction. So is the chloroform layer, which is expected to take up the sample, which is by partitioning principle from your aqueous phase, which is your plasma. So plasma, which contains your drug, once you shake it with chloroform, the actives are expected to partition into chloroform. In this case, definitely, as I said some time ago, that your physicochemical properties matter. So you must know whether your drug will partition into the organic phase. And if you're using this technique, that is the best I would prefer for HPTLC application later on, simply because we are aware that when I have some organic solvent in which my drug gets extracted, you always have the ease of application by the applicator in TLC, because then drying with the nitrogen flow becomes much easier. So in protein precipitation technique, we are aware there is always a dilution of the sample that happens, a bit of a drawback. But you go for liquid extraction, it rather gives you an advantage that you have this chloroform layer separated, transferred to separate tubes, you can evaporate it and reconstitute it. So when you are reconstituting it in the smaller amounts, you'll have the best advantage that you have concentrated your sample further. So then estimation using your any chosen technique, be it HPLC or TLC, will help you. So bioanalytical methods, HPTLC will be the technique of choice because of its high throughput advantage. Now coming to a popular research work that many of the M-Pharma students take up in their research semesters is development and validation of stability indicating methods. Now, I'm sure you must be knowing that stability indicating methods are simply the methods which are able to resolve any product of degradation and then help in quantifying the drug of interest even in presence of degradation products. So with this short test and sweet test definition, let's see some examples of how we have used HPTLC for stability indicating methods. I'll take you back to a work which was done way back in 2010. This was stress degradation of ofloxacin by HPTLC, and we had compared it parallelly with the technique of bioautography. I'm proud to say that this is a work of my UG students, Anuja, Karan, Sneha, and Venkatesh. Were a group of 4 enthusiastic students from UG who managed their time well. I must acknowledge my colleague Shivani Rao for microbiology work under bioautography here. And these 4 students, all of them are now in US pursuing their own research. And coming back to the work this team of enthusiastic students performed was ofloxacin. They exposed it to the typical stress conditions of acid-base hydrolysis, oxidation, photolysis, thermal, the usual ones. And then the first thing that they had to do was have a typical densitogram. You have to check. Now, the ofloxacin sample that we had, generally we obtain drug samples as gift samples. Now, this ofloxacin sample was showing us one more peak, though it was well resolved, so we didn't mind what it was. We use the bulk sample as it is, as we receive it. We do not go for further purification, so to say. So we notice this small shoulder peak. Probably it could be some impurity we inferred, and we continued with this. So this was the typical densitogram. And then when they exposed it to different stress conditions, a typical peak we would be looking for at around 0.5 RF. So this is for a typical linearity densitogram, as you can see. In bioautography, the same plate you have to place in contact with the agar plate, then incubate it. This plate already has the microbes which are inoculated. And when you go for you keep it at the temperature required for that organism to grow, you will see that this is the picture that emerged at the RF of the drug, that is ofloxacin. You can see a clear zone here. Now, this is something which indicates that ofloxacin from the plate has diffused into this agar plate, and this is the zone of inhibition. This is what was checked after every exposure to every stress condition. So here in the top plate, you could see different strengths of the standard. You could see different zones of inhibition. So this is something similar to the linearity in TLC. You could see a similar picture here. The only difference being that when we were to place it for bioautography on the plate, we had to cut the plate into small strips. So you could probably see that this is being marked here at the top, that the plate has been cut and then exposed. You can see the markings also. And at the RF of the drug, you can see the zones of inhibition. Now, once this is a routine linearity done, we have a linearity in bioautography also done. And then we have our sample exposed to different stress conditions. And here, as you can see on the label, thermal for 12 hours, neutral hydrolysis. So then we will spot the sample which has been exposed to different stress conditions on TLC, develop the TLC plate, then have it scanned under TLC, HPTLC scanner. And once my scanning is done, then we go for cutting the strips and putting it on microbiology, say agar plate, which already has the inoculum into it, and then incubate it for the next required period of time. and compare the percent degradation by HPTLC and under microbiological studies. This was a very interesting project we took up, and it was really a novel work that time. So we enjoyed doing this work, and I'm sure my students, as I mentioned, the enthusiastic team of UG students who worked on this, uh, I'm sure they could learn much more than what my research lab had to offer. It's the teamwork. So there is a great difference between UG and PG research work, that under UG research, it's generally a team of students who work together as a team. And in PG research is the individual student definitely who has to take up their own research project. Both UG and PG research have their own pros and cons. So here the teamwork has paid off. They could get a good publication done. Let me have one more example of stability indicating method. This is a work by Pooja, wherein she had looked for sofosbuvir and velpatasvir anti-viral drugs which are used for hepatitis C treatment especially. So we were lucky to get these drugs from my friend from NATCO, and then they could work on this combination. Pooja could work on this combination. The work involved development of initially a typical densitogram, that is optimization, to have these two well separated, well resolved. These two drugs in combination also have a ratio of 1:5, but still it didn't affect the resolution. You have sofosbuvir and velpatasvir eluting very well resolved from one another. The work involved the usual exposure to different stress conditions, and then such nice images she could get. Her work was, she was very good at her lab work. Just see the beauty of this image. You can see, even if I don't tell you what this image stands for, I'm sure you take good look at it and you will be able to infer that this image uh, is showing you, highlighting the benefit of high throughput. Here you could clearly see that first 6 tracks stand for linearity. You can see the peak area is increasing for this pink color. And then you have track number 10 to 19. These are accuracy studies by a method called as standard addition. Now we know, if we try to quickly recollect what we learn under accuracy studies, one of the methods to determine accuracy or confirm the accuracy of method is to go by a method called as method of standard addition, wherein to a fixed sample, as you can see, probably the first one is a blue colored, is a sample, which is analysis of a dosage form, to which you add standard amounts or known amounts of the standard. It could be 80%, 100%, 120%. You could see the increase in the height of the peaks that follow. Same is the case here. You have your sample and the standard additions. You could see the peak areas rising slowly, because what you have is sample 100%, to which you add 80, 100, and 120% of the standard. So standard addition method, the whole of accuracy study along with linearity, she could complete with a plate and high throughput quite visible here. You could see a nice track, and here, even if I don't show you the label, I'm sure you can guess easily that this is a image showing precision. We have the same concentration applied minimum 6 times, and you have to determine the area and try to find out whether the percent RSD, relative standard deviation, is within limits or no. So I think with HPTLC, such advantages matter a lot, that on the same plate, so many samples could be applied. And don't you think, if you submit such a nice image along with obviously the right results to any peer-reviewed journal, I don't think there is any doubt left They will be accepting your paper in no time without any queries, because the image is speaking for itself, as we say. So this is a work typically done under stability indicating method development and validation. Going ahead, we will have another application, which is applying the TLC method for dissolution method. Now, dissolution testing is a routine of the test in, say, or tablet as the pharmaceutical dosage form, capsules for that matter. And dissolution methods very commonly seen. You will find that most of the methods are by UV spectroscopy, uh, more so if we are referring to a dosage form which involves a single component. So the most preferred method obviously will be UV spectroscopy, where you don't expect any interference, so to say, from any other. Other common technique is HPLC, but as we know that there will be minimum 6 samples to analyze, and if you are talking about sustained release formulation, wherein you have to determine the dissolution after a typical time interval, then you will have HPLC, which will be running throughout the day probably. And then it comes to HPTLC, again, the advantage of high throughput and a renewable stationary phase. So HPTLC could be an option. Uh, let us also highlight a drawback here. I need not be only stressing the plus points of HPTLC, but when it comes to dissolution, one of the problem is there for HPTLC, that you will find that our dissolution media are aqueous. So when you want to have the spotting done directly from a dissolution jar going to my HPTLC plate, you know that water is not going to evaporate as fast as maybe any other organic solvent. So then my spotting speed has to be lower. Otherwise, you will find that the spots get diffused, and that could be leading to a bit of problem in development later. So yes, there is an alternative. There's a solution to the problem, that the speed of spotting has to be reduced. But yes, you could still apply this to dissolution technology. Looking at the time, I'm not going to go into detail of this paper, which my student Komal had taken up. She had worked on pipotaxin bisalate tablets, and she had developed a method for dissolution by all these 3 techniques, and then she had compared the results. But for lack of time, I'm not going into details of the paper right now. Before I say that, yes, this is a very good technique, we must also look at what are the probable errors that can happen. And once you know what are the common causes of errors, let me put it not as a mistake, but some errors, we can avoid time wastage. So let's look at some common errors. Say the common errors could be the way you prepare mobile phase and then transfer it to the development chamber. It's not a good idea to have mobile phase prepared using a measuring cylinder, because you know that typical mobile phase volumes are to the tune of 10 to 20 ml. So if you're preparing a 10 ml mobile phase and you are using a measuring cylinder, a slight difference in the meniscus could lead to a lot of change in the actual volume that gets delivered. So it's a good idea to use a pipette. Prepare a mobile phase in a small iodine flask using a pipette to add 1, 2, 3, whatever components you have in a mobile phase. Shake it well to have a good homogeneous mobile phase. Then pour it into your development chamber from or on to a filter paper, which you put on one side as an aid to have chamber saturation. And I'm sure as you are attending this webinar organized by Anchrom, I'm sure you have looked at so many of their videos on their website, wherein they tell you about how it is to be done, what is the best practice of using an HPTLC. And if you haven't done it, you can definitely do so in your free time. That preparation of mobile phase and then transferring it to the development chamber has to be done in a correct technical manner. Please do not underestimate the importance of saturation time. That also has an impact on the RF value. Many times, the pre-coated TLC plate that we handle, ultimately it's an aluminum plate. So if it gets overheated, if you keep it in oven at 60 degree for longer than expected, probably there are chances that it will get curved. And if you place such a plate for development, there could be a problem of different RF values on different tracks. So avoid doing that. Avoid overload. In the beginning, in the initial trials, we may go for a higher concentration of sample to be applied till such time as you're not sure of what is the RF value, which peak stands for the peak of your sample and so on. That is in the very, very initial stages, probably higher concentrations are okay. But later on, avoid overloading the plate. This will certainly affect the resolution. Spotting speed is something of concern, as we saw just now, that when it comes to spotting of aqueous samples or hydroalcoholic samples tend to go for a reduced speed of application. Our CAMAG applicator does have an option of changing the application speed. So distance between tracks. Yes, let me highlight this. This is because looking at the advantage of high throughput screening, we many times tend to go overboard. We tend to try and spot many samples on the same plate, and then we tend to overlook that what is the bandwidth which is ideally expected. So 8 mm, 6 mm, but if you go below that, 4 mm, 3 mm, your accuracy is going to falter. So distance between tracks will go on reducing as you go on trying to accommodate too many samples in one plate. So do not compromise on distance between tracks. The size of development chamber matters. So if you have the same plate developing in a 10 by 10 chamber or a 20 by 10 chamber, please do keep in mind that your RF values can vary. So try to maintain the chamber size as you go for optimizing and validating. Band length, as we discussed just now, does matter. So yes, apart from the applications that we have tried in the lab at AISSMS College of Pharmacy, I have tried to showcase some through the open access papers of some of the students. I have told you some points of precaution, how to avoid the common errors. And definitely, I would like to acknowledge with gratitude the help and support, encouragement by the principal and the management of AISSMS College of Pharmacy for providing this research facility, which is always in a maintained condition. Why are we highlighting this? Is because without an AMC for such sophisticated instruments, I mean, we cannot expect to have an instrument and not have it working all the time. So that's a great support from principal and management. I definitely appreciate the work of my students, whom I have shown some representative papers. And I definitely acknowledge the technical support and routine maintenance support, which is provided by the whole of Anchrom team. I will have lots of praise for them for timely help, because that's what has helped all of the students to be able to complete the research work in time. Because we know in academic research, there are deadlines for thesis submission, project report submission, and without timely help from Anchrom, this generally may not have been possible. So I will thank all of you for your patient listening. And this is the image of my college at Pune. So all of you are most welcome to visit us. And this is the lab at the corner. This is the QA lab where we work, and we have our HPTLC here. So thank you so much once again. And I will be happy and I will try to address if there are any doubts to the best of my ability.
Thank you, ma'am, so much for this wonderful presentation. I think ma'am has not left any corners of the research which is there pertaining to the pharmacy concept, and very practical and straight. We saw some very good experiments done from onion and different aspects of force degradation, and then how it has been collaborated with effect directed analysis. So a lot of interdisciplinary research, and that is what we talk about when we are researching something at very core fundamental level. So I think ma'am has given us a wonderful insight, and this clearly shows that we have the right speaker for today's session, and we have a fantastic knowledge increment. So now I will just go through some questions, but we just received one question. A person, a madam has asked if any technique in HPTLC is there for checking the authenticity of the seaweed. Now, it is not directly related to this topic which madam has taken in, but if any information, ma'am, you would like to share, or we can go ahead with, I can answer this question. Um,
You can, sir. I have no
yeah,
idea about this, frankly.
okay. So seaweeds are generally correlating to your again the plant kingdom, and that is why we have definitely few methods. Now, what you are trying to see in this plant is very important. You have some pigment compounds which are of crucial importance in such kind of seaweeds, and secondary metabolites. So, if we are specific to know that what kind of a specific metabolite you are interested in, a method can be adopted, and routine quantitative estimation can be done. So absolutely no problems with that. And I will definitely, we will send you some literature papers which are available on CPED to give you further insight on the topic. Now, um, we have some things on the chatting. Let me check if we have a question over here. Um Okay, so we don't have anything over here. I think only one question we had, and so we will quite summarize the session. We got a detailed understanding of different pharmaceutical aspects and investigations when a formulation is created, right from the original molecule, the raw material, to the finished product, and at each stages when it has been subjected to quality control steps with the help of HPTLC, and going one step ahead to find out other possibilities of impurities and their functions. Dissolution studies, madam has stated upon, and there are certain restrictions how dissolution studies can be restricted. Okay. Uh, Okay. One more question just now we have received. We'll quickly take up that question. There's a question, ma'am. Somebody asked us that can we carry out characterization of novel herbal formulations by HPTLC?
Yes, surely so. So, as I have been stressing that for herbal research, HPTLC is the best technique that I would recommend. So, for her new herbal candidate that you have with you, probably you could go ahead and fractionate it with different solvents of different polarity, and try to go for the TLC pattern first. So, as we call it, TLC fingerprinting could be the starting point. And from literature, if you come across the typical actives which are known to be present in it, if you could isolate or you could procure from market any typical marker which is active or unique to that plant, I mean, that's the best way to go ahead and standardize it. So TLC fingerprinting could be the starting point, and then quantification of a typical marker from it could be the next step in your work.
Thank you, ma'am, for this insight. For anybody who has a question, we can take up one or two. So you can raise your hand. We will unmute you, and you can live ask some question. So that's a wonderful question which we received about the characterization. And I think, ma'am, TLC MS interface has not been still explored, and that can be a wonderful possibility because many other places we have seen that TLC MS interface is becoming an interesting tool to, not only just find out the MS characteristic, but as an isolation kind of system where you
Very
quickly
much.
isolate that spot and analyze it using different techniques.
Right.
So,
Very true. As you correctly
that,
put, I think I
huh,
had said only that confirmation of identity I had shown just by the RF value and the UV spectrum of the sample. But then, yes, as the technology is evolving and we have that TLC MS interface coming up, that's the next step. And surely, if the MS pattern is confirmed, there is no other doubt left about the identity actually. So that is
sure.
something we can, and yes, I know that though not every lab will have that TLC MS interface, Anchrom is always coming forward to help the research students. So I think we
Hmm.
can, all the students are anytime welcome at Anchrom to do such a work. And that's the great work Anchrom team is doing.
Thank you so much, ma'am. And we always, look forward for such kind of young researchers who are coming up and they want to really do a good quality work. So Anchrom is always open to this. And ma'am, we have got one another question. Can we quantify the marker compounds from uh, the same formulation? Um, basically, he is asking about quantification of markers from same formulation. So I think that madam has already answered. We can obviously do a marker study if you know what marker you are looking forward based on from whether it is your API, that is a active pharmaceutical ingredient, or material which is require coming up from the natural resources or botanical origin. You can definitely go ahead. I think it is quite possible. So I think we have taken up all the questions. And Rahul, do we have any raise hands for asking question in the participant window?
No, sir.
Okay, great. So we will then, I think I'll present the vote of thanks then, and we'll summarize the session, end this session. So on behalf of entire Anchrom team, I especially thank Damle madam for taking out her valuable time so much and briefing us with this wonderful insight of using HPTLC in the perspective of academic as well as also not leaving the concept of industry core relevance, because that is very much important. We do a lot of work, but then it becomes an bookshelf work, and it stays over there and it does not become of any importance. But what we can see is madam has addressed two very crucial points of pharmaceutical analysis, and that is how she has been uh, also explaining the concepts of HPTLC. I really thank her for her valuable inputs, and it definitely shows the experience how person has got into depth, and also with the brilliant team she has, wonderful results we have got. So I again thank you very much, ma'am, for joining us. I also thank your management for the continuous support which they have given, and to entire not only uh, uh, um, in keeping the instrument, but to us also, so that we can help your system to keep it at all time very best, to give all researchers hands-on experience. So, with this note, I would like to now hand over the session to Rahul, and we will now write to. If you have any questions, please write us at lab@ancrom.in, where we will reply in one working week. Uh, that's the maximum time, and hope you all had a wonderful session. Again, special thanks to ma'am for accepting our invitation and interacting with the participants. So thank you so much, ma'am.
Thank
With
you.
this note, we will now end this session. Rahul, ma'am, with your permission, shall we end this session?
Thank you. Thank you so much.
Thank you
Thank you,
so
ma'am.
much,
Thank
ma'am.
you.
Thank you so
Thank you.
much.
So all of you take care, stay safe, and stay tuned to such lectures which Anchrom is organizing with really the experts in this field, and it feels really special for us because these experts have been into a lot of experience, and there is no supplement to gray hair, I always tell, and that is why it's wonderful always. So thank you all. With this note, we'll end this session. Rahul, I
Yes,
request
sir.
you to end this session.
Yes, sir. I'll end the session. Thank you so much, madam. Thank you all the participants.
Thank you.